All right, ladies, this video is going to get us started in one of our more challenging topics, I think, which is cell communication and cell signaling. I feel like something that we take for granted in biology prior to college is that cells have a really complex way of communicating with each other. It's the reason that your body works as well as it does to do all of its different jobs. And in freshman biology or other classes, we just kind of say, hey, this happens and this happens. But in reality, there's a lot that goes on between that first thing and that second thing. And that is what cell signaling is all about and what we're going to get into with this topic. So on that first slide, you see a frightening day for a gazelle, right? Is when a predator shows up. The point of that being that every response in the body is the result of cell-to-cell communication. So you guys have probably heard of the fight or flight response before, right? You get in a stressful situation, and your body reacts in certain ways that either make you more prepared to defend yourself or more prepared to run away, right? And that's why we call it the fight or flight response. This is the result of hormonal pathways in your body, right? Hormone signals released by one group of cells after you detect that stressor and received by another group of cells that then lead to some kind of reaction. The hormone we're talking about here is epinephrine, or sometimes also referred to as adrenaline. So something that I'm sure you guys have heard of before. And epinephrine or adrenaline is released by your adrenal glands, which is right above your kidneys, and flows throughout your bloodstream and causes different responses in different parts of your body. Now, this is just one example of something that I figured you'd heard of before, of how cells can talk with each other. Now, usually when we're talking about cell communication, we're talking about chemical signals, right? Chemical signals that are released or secreted by one cell. So that might be sort of a newer term for you guys, but right, secretion would be release from a cell, and goes out and targets another cell in the body, which could be really far away, right? And one thing that's cool about these signaling pathways that involve these biochemicals is that a lot of times the pathway is really similar in all sorts of life. So there's sort of like this evolutionary connection between us all in the ways that our cells communicate with each other. Now, in most situations, we have an external chemical signal, a signal that's outside the cell, that leads to an internal series of steps that we call a signal transduction pathway. A big mouthful, I know, but transduce or transduction means that we're going to have a little bit of a change in the type of communication. So as we'll talk about here in a minute, you probably have some kind of receptor, right? So you got this receptor that binds to the outside of a cell, and then inside you see some changes that take place. But the receptor itself does not go inside the cell. It stays out here on the outside. Now, signal transduction pathways in cells are used to coordinate all sorts of stuff. We could come up with literally hundreds, if not thousands, of examples of this in a complex organism like us. But basically, any kind of physiological function that you have is controlled in part by signal transduction within cells. A couple of examples your textbook brings up. One is that bacteria actually do this. Bacteria do something called quorum sensing. And quorum sensing is where bacteria, which again are just a single cellular organism, they release a chemical into their surroundings, and they can detect those chemicals in their surroundings. So in other words, the growing colony can sense how many bacteria are around them. And this might lead to increased reproduction or decreased reproduction by those bacterial cells, ultimately trying to form a covering, which is what you see right here in this picture on a surface. And we call this covering a biofilm. Biofilms can grow all over the place. If we're talking about a pathogenic organism, biofilms can be really dangerous. But probably for you guys, the most common biofilm you can think of is if you think about like falling asleep without brushing your teeth, and you wake up and you just kind of have this filmy nastiness on your teeth. What you're really feeling there is a biofilm of bacteria that's grown on there. You might also just feel that throughout the day if it's been a while since you ate or brushed your teeth and things. Now, eukaryotes, like us, we also have all sorts of signal transduction things going on. Nothing quite like quorum sensing, but one thing a lot of times that scientists talk about are circadian rhythms, which you've maybe heard of before. Circadian rhythms is sort of a concept that you have this 24-hour clock running in your body, and you reset that clock basically every day when you go to sleep and get back up, or when it gets dark and then it gets light again. But your cells will actually maintain a roughly 24-hour rhythm even without those external cues. It's not exactly 24 hours, it's actually a little bit longer. It's kind of almost closer to 25 hours. But for instance, if you have a group of miners that are underground in a mine for a few days, they'll actually still be able to kind of know what day it is and whether it's morning or night, even without that external stimulus of light to kind of reset their rhythm. And that's, again, because of just these different signals between and within cells. Now, what are the different types of signals that can go out? Well, the first thing we want to look at is called a local regulator. Local regulator, as you might guess from the name, is a signal that just affects cells right around it. So the first option, which we're really not going to talk much about at all, is cell-to-cell contact, right? So as you can see up in this picture, sometimes there are actual just connections between cells, like you see here in both a plant and an animal example. Other times there's cell-to-cell recognition, which takes place outside the cells. There's no opening between them like you see above here, but it is actually one cell contacting the other. So there's no real release or secretion of a chemical here at all. Now, the second option, which we'll see quite a bit this year actually, is called paracrine signaling. Now, paracrine signaling is where you do secrete or release a chemical, but it stays right in your immediate vicinity. It only affects cells that are very nearby, like you can see here. So most of the growth factors in tissues are paracrine. Last time in class we talked about platelet-derived growth factor, or PDGF. That's an example of something that acts in a paracrine way when a platelet releases a chemical that just goes to that other kind of cell nearby. Third option is a really specific type of local regulation, and that's synaptic signaling. And this is only seen in neurons, right? Nerve cells. So when a nerve is stimulating a target tissue, whether that's another neuron, whether that's a muscle, whether that's a gland, what happens is the neuron basically forms this connection chemically with the next cell. And it does this by releasing what's called a neurotransmitter. So here in this picture, these little red dots are these neurotransmitter chemicals, which are released by one neuron and then received by some kind of target cell. And we call this synaptic signaling because a synapse is this little gap here between the neuron and the target cell that they have. We'll see a lot more about this later this year. All right, now the other option, which really is going to be more of our focus for now, is long-distance signaling. And this is mostly dealing with hormones. So in the human body, we call this endocrine signaling. So I know you've heard of the endocrine system before. Oops, I just noticed this picture's overlapping here. But the endocrine system is a system of glands which can release hormones. And hormones are just chemicals, oftentimes made of protein or lipid, which move through your circulatory system. So they move through your bloodstream, and they go to target tissues. Now, the thing about endocrine signaling is a lot of times the hormone you release is going to travel all through your body and far away in your body. It doesn't need to be nearby cells. So, for instance, when you get that release of adrenaline from your adrenal glands, and those are located by your kidneys, so we're talking kind of your lower back. But that adrenaline is going to affect your brain, and it's going to affect your muscles throughout your whole body. So these signals can really get anywhere. Now, plants actually have an endocrine system as well and release hormones that we'll learn later this year. A lot of times plants use gases as their hormone. So you might have heard of ethylene before. Probably, if you've heard of any plant hormone, that's the one you've heard of. And that leads to a lot of different things, but particularly ripening in fruits and vegetables. So you may have heard that before. If you've got some bananas and you got them sitting next to some other fruit, the other fruit might actually ripen quickly along with the bananas because of ethylene that's being released in the air. Now, in animals like us, you've got a lot of different chemicals that are released to your bloodstream and function as hormones. One example, again, I'm guessing you've heard of, is insulin. Insulin comes from your pancreas and helps to lower your blood glucose levels. And so after you eat, blood glucose levels go up, and insulin helps to bring them back down to normal. So this slide is a big deal. In terms of the AP people, we're certainly going to learn some details throughout here, but the most important thing to take away when it comes to signaling is this right here. And that is that the general way in which our cells undergo signaling is a three-step process. And this three-step process consists of reception, transduction, and response. Now, the remainder of these notes that are going to take us a couple of days in class are going to go through each of these steps in lots of detail. But the general idea you have to know. And so that is this. First, you have to have reception of a signal. So there must be some sort of signaling molecule that binds to some kind of receptor. And we'll add a couple new terms here in a minute to that. Then we move on to transduction. Right? A signal transduction pathway, or sometimes called a signal transduction cascade, where you've got various relay molecules which pass this information on down almost like a chain inside the cell. And then finally, step 3, response. The cell responds in some way. This can be really wide-reaching, right? This could mean a lot of different things. And so we're going to slowly and carefully go through each of these pieces: reception, transduction, and response, so that hopefully you have a good feel for how all this works. All right, so we're going to start with reception. Now, the first sentence here is a great overall summary of what we're looking at, and it comes back to something that I told you a while back would never go away, and that's shape. Right? So a signaling molecule is going to bind to a receptor protein, and that receptor protein is going to change shape, leading eventually to some sort of transduction and then reaction by the cell. So receptors are essential. All right, you've got to have receptors on the cell to bind with some sort of signaling molecule. Now, here's the cool thing, right? You've got a ton of these signaling molecules in your bloodstream, but they only affect certain target cells. And the best analogy I ever heard about this is actually in your textbook as well, but I heard it back in college. And that is that your body is almost like a radio. Here's something weird to think about. All the different radio stations, right? Radio waves, they're all around us right now. Right? There are all sorts of these little energy waves, these radio waves, all around us right now. But You don't hear them, right? I don't think. I don't know if anybody in here is different than me, but you don't hear radio stations just in your ear. So the first thing is you have to have a radio, right? In order to catch any of those sound waves, let me see how poorly I can draw a boombox here. You have to have a radio, right? But you also have to have that radio tuned to the correct station. So it has to have some kind of antenna, right? And it has this antenna, and then you set that dial to whatever station it is that you like. So I don't really know local radio stations at all, so I'm going to say 96, right? Or it's like 96.1 or something like that. So you have to tune your radio to 96.1, and then you will hear, we'll say it's this red radio wave, right? But the other radio waves are out there. Now, your body works the same way. You've got all these different signals, right? All these different signals in your bloodstream, all these different signaling molecules. But the only time that you will actually detect them, right, is if you have a receptor for that. If you tune your cell to that station. So these receptors are critical because they will appear on cells that want to accept a signal and respond to a signal. Now, big vocab term here, ligand. A ligand is any molecule that specifically binds to another molecule. So a ligand is actually kind of a generic term, right? It's anything out there that will bind specifically to another molecule. So when we talk about signaling molecules, we rarely use that term. Moving forward now, we're going to use the term ligand. So a signaling molecule floating around in your bloodstream is called a ligand, and a ligand will bind to specific receptor proteins on or inside your cells. So again, signaling molecules are ligands. Ligands bind to specific receptor proteins on your cells. Now try to help me remember next class. It's time for a video break. Pearl here is a fantastic video that you may or may not have seen before. And so next class we'll be sure to watch that so that you can appreciate little Pearl here, who kind of reminds me of my own kids when they were a little smaller. But receptors, if you notice at the end of that last slide, can be on or in cells. Well, that's kind of weird, right? Generally, we think of them as being on the surface, right? And these would be receptors that are extracellular. They're stuck in the membrane. You guys saw this when you looked at the membrane a week or two ago. And these will look for ligands that can't pass through the membrane. And so in this case, we're talking about a ligand or a signal that is a big molecule. It's a polar molecule, something like that. And so you're going to have a receptor on the outside of the cell that ligand can bind to. Now, some receptors are actually inside of cells. And the way this works is in this situation, your ligand can pass through the membrane. All right, so can pass through membrane. So they're small, they're non-polar, they're hydrophobic. Think of things like gases. Right? In this case, the signaling molecule goes right through the membrane and it binds to a receptor inside the cell. And we'll look at an example here in a minute. Now, we have to cover 3 types of extracellular receptors. So there are 3 kinds of receptor that we generally can see on the outside of the cell. And each of these 3 types has tons of subtypes. For instance, this first one, there are like over 1,000 different kinds of GPCRs. But we're just going to talk about it as a general group. All right, so the first type of extracellular receptor is called a G protein coupled receptor. Now, these are super common. These are probably the most common types of extracellular receptors we see in the body. As you see this year, as we get to more and more examples, a lot of times things start with a G protein coupled receptor. Now, these are made of 7 pieces that are stuck in the membrane, and they work in a very predictable way. So we've sort of got it in words here, and then we've got it in pictures on the next slide. So first thing that happens, you have a ligand, right? Ligand binds to the receptor. You get a shape change, like we said a minute ago, in the receptor, which causes it to bind something called a G protein. G proteins get turned on or activated with GTP, which is basically just like ATP, so this is a form of energy. The active G protein moves over to another enzyme, activates it, and you get some kind of cellular response. Easy, right? Okay, let's look at this with the picture. So you've got this G protein coupled receptor right here lodged in the membrane. You got this thing called a G protein, which right now is inactive. And then you've got some enzyme that's waiting to be activated over here. So first thing that happens, signaling molecule, aka ligand, binds to the receptor, activates the receptor. The receptor then changes its shape a little bit, kind of like right in here, right? Which allows it to bind with this G protein thing. G protein gets activated. When it does that, it can move. So if we look over here at box 3, it can move over and activate this enzyme, right? Now that thing's active. And that will lead to some sort of cellular response inside the cell. And then after that, everything resets, right? So it can be used again. So again, ligand binds receptor. Receptor activates G protein. G protein activates another enzyme, which leads to a cellular response. Why don't we even just put it over here, right? So ligand binds receptor. Receptor binds and activates G protein. I know you guys love my handwriting. G protein binds and activates another enzyme. And again, there are like over 1,000 different types of G protein coupled receptors. So they can be involved in hormone pathways. Like it says here, they're involved in your senses and your ability to sense things. They're involved in development and genetics, but all of them work in this predictable pathway. There's just different types of receptors and enzymes and stuff that fill these little roles. All right, second group, not nearly as common as G protein coupled receptors, are something called receptor tyrosine kinases, or RTKs. Now, first off, here's this term that we've seen a couple times this year, kinase. Remember, a kinase in general is an enzyme that phosphorylates. So it adds phosphate groups to stuff, right? So these are a type of receptor that has this ability. So an RTK is made up of two monomers. When you bind to two ligands, they join together, these pieces, to form what's called a dimer. And this dimerization of the receptor makes the kinase part active. So it adds phosphates actually to itself. And then once that happens, the receptor is activated, and it can lead to actually several cellular responses, which so makes these really unique. A G protein coupled receptor gets turned on, leads to one response. An RTK gets activated, it can lead to several different responses. So here's what this would look like. All right, so it starts out as two pieces here. All right, two different monomers. Once we bind to two ligands, it allows it to dimerize and just come together to form one piece that has two parts, hence dimer. Once that happens, you can use some ATP and you can phosphorylate this bad boy. Once it's active, you can set off several different cellular responses. They show two here in this picture. It could actually be more than that. All right, it's a 2+. You could get cellular responses going from other areas here too. So essentially the binding of one type of signal can lead to several different responses inside the cell when it comes to an RTK. Last thing here, ligand-gated ion channels. Now these are not something that we're going to talk about as much as the other ones, but these are essentially things that you've already learned about with the membrane over the last couple weeks. Right, you got channels, and some of these channels are gated so they can open and close. Well, on some specific cells, they're ligand-gated ion channels, which means when a ligand binds to the channel, then it opens up, right? And then it releases the ligand, and it closes back off. Generally, what we're talking about is opening a channel so that some kind of ion can get in and out. So whether it's sodium or potassium or chloride, something like that. And where we most often talk about these is in the nervous system. So again, when you have a nervous signal across the synapse that we mentioned a little while ago, and it's received by that target cell, what usually happens at that target cell is this. All right, so these neurotransmitters, which is what this would be, here, neurotransmitter comes in, binds to the receptor, opens up a gate, and causes the influx of something. And in neurons, what we actually end up seeing is an influx of sodium. This influx of sodium activates the next cell, and the signal kind of moves forward. Now, just to tie this to real life for a second and tie it actually back to our previous class, there are several types of cancer now that have been identified as having these cells that have malfunctioning RTK receptors. And one of those specifically is this HER2 receptor that we mentioned at the very end of our last set of notes on breast cancer cells. Basically, a HER2 receptor should be on those cells doing a normal thing. But if it's malfunctioning, what you'll see is hyperactivity, right? Too much activation of this receptor, which then means too much of something in the cell. Now, this used to be a bad thing. If breast cancer had a high malfunctioning HER2 count, that was bad. That was a very aggressive form of cancer. But today, because of that medicine we mentioned last class, Herceptin, this is highly treatable because Herceptin will bind these crazy active RTK receptors and basically shut them down, lock them down, so it inhibits cell growth and slows the formation of tumors. Last thing for now, I know this is way enough for now, is that we also do have, as I mentioned a few minutes ago, some intracellular receptors, some receptors inside the cell. And these work as long as your ligand can get across the membrane. All right, so if the ligand, like you see in this picture, can cross the membrane, then we can have a receptor inside the cell for that. Any of your steroid hormones, which you can think of that like your sex hormones, like testosterone, progesterone, estrogen, these can have internal receptors. The thyroid hormones can have internal receptors. Gases can have internal receptors. So we use nitric oxide for some things in our body. Best example of this, at least in terms of what scientists understand, is testosterone. And that's what this picture is essentially showing you. So testosterone can, be released into your bloodstream, moves to a target tissue, actually goes into the cell where it binds to the receptor and forms this little complex. And then from there, testosterone receptor complex actually goes into your nucleus and increases transcription. So the conversion of RNA to DNA. The conversion of DNA to RNA. I know we haven't really talked genetics yet, but hopefully you remember that vaguely from freshman year. So this testosterone receptor complex actually acts as a transcription factor. It increases the expression of certain genes, expression of genes in your tissues. Okay, I think that's more than enough for now. And it takes a look at the basics of cell signaling, followed by the first big piece, which is reception. So coming up in class, we're going to look at the second stage, transduction. We're going to look at the third stage, response, and try to put it all together. So thanks for listening.